we would be studying on ascent of SAP, that is how water moves up on trees, translocation, how foods which are manufactured from the leaves pass down via the phloem, and as well as transpiration, that is what happens to excess water which is lost. Now all these three concepts are associated together, and together they form the sort of a concept termed as plant water relationships. So along with osmosis as well as along with imbibition, the chapters are integrated. Now we are going to read the chapter in terms of certain very important concepts. Concept number 1, what happens to the water and ions? When we take water and ions, they are absorbed. Primarily the pattern of taking is termed as absorption. They are absorbed only when there is an increased surface area. So the increased surface area is attributed to A, root haze, primarily because of the presence of root haze there is absorption of water and ions, and 2, there could be lateral roots. So supposing we have a plant which has better lateral roots and root haze, that means the absorption efficiency is very high. Now what happens in situations wherein we do not have the roots, whereas there is uptake of phosphorus? Uptake of phosphorus is enhanced, or phosphorus utilization is enhanced on mountainous regions where the nutrients are less due to the presence of mycorrhiza. So mycorrhiza helps in phosphorus utilization. So root haze, lateral roots and mycorrhiza together contribute to increased surface area for absorption. So increased surface area for absorption, especially in terms of water and ions, is attributed to these three very important sort of concepts. Now we are looking at what happens to transpiration. Concept number 2 relates to transpiration. Transpiration increases or it decreases. Now transpiration increases with high light intensity. So light intensity is very, very important here. The higher the light intensity, higher is the rate of transpiration. Same is the case with relation to temperature. The higher the temperature, the more is the water loss. And parallelly, even we have, in case we have plants which are subjected to high wind velocity. So higher wind velocity, higher temperature and higher light intensity means even the transpiration rate goes higher up. While if the humidity gradient if the humidity is the same inside the cell as well as the outside, if it is just normal, then transpiration drops. Humidity gradient is the same, means transpiration will drop. And again, in case there is more abundance of carbon dioxide near to the sort of a stomata, high carbon dioxide at stomata region. So there is competition between carbon dioxide and water, and thereby water is not lost and carbon dioxide is sucked in, thereby the transpiration rate is low. So it's important that we keep into consideration these factors: humidity, carbon dioxide, wind, temperature and light. What happens in the case of transpiration? How does it go high and low? Now we come primarily to a very important concept on how SAP flows, ascent of SAP. Ascent of SAP in plants. So, here if we look at the driving force, we have a driving force, especially in terms of bulk flow. Then again we have the location where it is present, and third we have the pressure potential of the SAP. If you are going to look at these three attributes, number 1, what is the driving force? 2, what is the location? 3, where is the pressure point? And we are looking in terms of xylem as well as we are looking in terms of phloem. The driving force in the case of the xylem is the transpiration of leaves. And again, when you take the location, where are they located? Especially when you take xylem ascent of SAP, it is located in the non-living vessels, that is vessel elements and tracheids. That is the dead tissues primarily help in ascent of SAP through the xylem. And the pressure potential of the SAP, how is it? It is primarily, it is negative. Why is it negative? Because there is a pull from the top. And thereby, because there is a pull from the top, it creates a tension. So, it is very important that we understand the three important concepts related to xylem and well as even when we come to the phloem. When we come to the phloem, the driving force of the bulk flows is primarily due to active transport of sucrose. The active transport of sucrose occurs at the point where it is manufactured, that is the source. So, you call it as at source. Then B, we have the location, it is present in the living tracheids. And 3, the pressure potential of the SAP is due to a positive push. It is positive and it is because of a push. And the push primarily it creates push from, where does the push occur? The push occurs from the source, from source. And because it creates because it occurs from the source, it creates a sort of a pressure. So, here we could understand sometimes it is also bidirectional. Now, when you look at the ascent of SAP, these are the basic points from which you could be getting the different sort of multiple choices. They could ask you what is the driving force, where is the location, where is the pressure potential of the SAP, what is meant by tension, and what do you understand by means of pressure. Into both active absorption of ions as well as carrier concept. Now, what is meant by active absorption of ions? Active primarily refers to utilization of ATP, and it has been studied as implied by van der Honert. So, supposing we have an ion and the ion is activated, When you say an ion is activated, it means it is charged. This particular ion is added to a carrier protein. The carrier protein is a big bus, and the carrier protein carries the ions. The carrier protein carries the ions and it forms an ion carrier complex. So, first one we have the ion which is charged, second it forms a carrier protein complex. This ion carrier complex shuttles between the outer free space. This is called the outer free space. The outer free space is also called the apoplast. So, the carrier concept can come over here and it can move via the sort of an apoplast and move through the sort of a symplast. It can carry the ions, it can shuttle between the outer free space, and this is termed as the inner free space. The inner free space is present within the cell. Now, whenever the ions move, active absorption of ions, it is against a concentration gradient. So, we have a concentration gradient here. This is termed as concentration gradient. There is already there is traffic. There is traffic of cations and anions from the outside to the inside of the cell. Various molecules like sodium, potassium, calcium, magnesium, etc. they keep on travelling. So, we have a cation load as well as an anion load with influx and efflux across the membrane. But a carrier concept is a special shuttle bus. A carrier concept is just like a revolving door. So, the carrier, what it could do is, the carrier molecule is a protein molecule which can selectively take up ions, and by means of they can be inflow as well as outflow. It could be influx or efflux, which is created by the means of the carrier molecule. So, in order that the carrier molecule moves through from the outer free space to the inner free space, it requires energy. And the energy which is required for the carrier molecule is ATP, and ATP is primarily derived from respiration. So, what are the main features of active absorption? Ions are free, ions are picked up by carriers, carriers go to form a carrier ion complex, carrier ion complex moves from the outer free space to the inner free space. It moves against a concentration gradient, and next one is, for the movement of the carrier molecule there is a requirement of energy, that is in the form of ATP, and ATP always occurs through from, or it is obtained from, the process of respiration. Next, we are coming specifically to carrier concept. A carrier is a protein. A protein molecule. Normally it could be a lecithin. Otherwise, it can also bind to form, it is made up of a phospholipid. So, you call it as a lecithin phospholipid. It could also be a cytochrome. More oftenly, a carrier molecule along with cytochromes. That is, you have a scheme of chemicals which can act both as an oxidant as well as a sort of a reductant. It can go to form a sort of a cytochrome pump. And there are, when you take the carriers, there are separate carriers for anions and also separate carrier for cations. So, when you take a carrier, in principle they are proteins. Common examples are lecithin or phospholipids. They can also go to form cytochrome pumps. There is a separate carrier for anions and a separate carrier for cations. The carrier concept has been proposed by a scientist whose name is Lundegaard. So, he believes, according to Lundegaard, a carrier concept or the carrier molecule is one which is involved with relation to utilization of ATP. And here it relates to specifically anion absorption. Anion absorption by utilizing the ATP from the process of respiration. So, source is ATP, movement is anion, proposed by Lundegaard, concept is a carrier. So, it requires ATP. ATP is a very important requirement for a sort of a carrier concept. Now, you primarily understand what exactly is active absorption of ions as well as carrier concept. Now, we are coming to a very important concept about ascent of SAP that relates to an osmometer. In an osmometer, we have a flask A and we also have a flask B. Now, flask A and B are attached by means of an upper capillary tube. This is a capillary tube. Then, in addition to this, both of them are connected laterally by a lower rubber tube. So, this is a plastic glass tube. Parallel to this, we have in A, we have a dialysis tube. As well as the same is case even in the case of B. So, this is a model of an osmometer. Now, when you look at flask A, in flask A, the flask A has water H2O and the dialysis tube has sucrose and dye. It could be having sucrose and dye. Now, once what happens is from the side tube we have water entering here, because B is only made up of water. From here water enters down here. Once the water enters into A, the sucrose and the dye move upwards into the capillary tube. So, water entry and the sucrose and the dye, they enter through the sort of a side tube. This is what happens in A. Now, when you take flask B has got water and the dialysis tube has water only. So, many times in a sort of a multiple choice, they could be asking you what is present in the dialysis tube in flask A. It is very important that we exactly understand it is primarily sucrose. Sucrose and the dye. How do you track the movement of sucrose? You track the movement of sucrose due to the presence of a sort of a dye. Continuing on the analogy of an osmometer, we understand that even a plant acts as an osmometer. Because we understand that flask A is considered a source and flask B is considered as a sink. Now what happens in flask A? The flask A, the source, it is just like the sort of leaves which manufacture the photosynthetates. It is similar to that of the dialysis tube. So here we have water movement into flask A, that is by means of osmosis from A, that is the water column outside, into the dialysis tubing. With the dialysis tubing has got sucrose and dye. So water moves inwards from the outer shell into the dialysis tube. That is what primarily occurs during phloem loading, that is during at the source where the food is being manufactured. Then the source and the dye move through the upper tube. The upper tube is typical of that of the phloem. So through the upper tube, that is the phloem, we have the movement of the sucrose and the dye, and thereby why is the movement? The movement is because of hydrostatic pressure. So we understand in flask A we had water, there is entry of water touches upon and pushes sucrose up, and this entire process is termed as hydrostatic pressure. And similar is the case with relation to when water gets loaded out into photosynthetic leaves. The photosynthetase in the particular leaves are loaded into the phloem, and through the phloem it just goes out. Flask B is analogous to a sink, that is water is forced out of B, as you understand in the analogy. When you take an osmometer, water moves out and the water returns via the tubing at the bottom. So the bottom tube which connects is analogous to the xylem. So xylem is always a tissue where there is return of water, and phloem is always a sort of a tissue in which there is movement of sucrose. Thus we say that a plant is also a sort of an analogy for an osmometer. Now why is there the flow of water between two ends of the system? Now sucrose dye solution flows through the capillary from A to B. So how is the flow of water? The flow of water is always from the left to the right, that is from A to B. Now due to flow of water, so we understand first there is flow of water, then due to flow of water, what really happens between the two ends of the system? The system will come to equilibrium. It's important we understand the statements like this. The system will come to equilibrium, the flow will cease when the sucrose concentration in A and B are equal. Supposing the sucrose concentration are equal, then only the system will stop. In plants, there is continuous flow, and the continuous flow is maintained because sucrose is added continuously in A. That is, leaves can photosynthesize continuously, and there is always withdrawal at B, that is from the bottom, from the roots always water gets pumped in. So it is a sort of a continuous process which occurs in plants, thereby there is flow of water. So how do you understand the source and the sink relationships? Because of the presence of a source and a sink relationship, we have an osmotic gradient. Now when you take an osmotic gradient, because of an osmotic gradient, there is flow of water. Now it is very important that we look at what is happening at the source. At the source, what is the source? The source is a transporting organ. What could be a transporting organ? A mature leaf or a storage organ which normally contains sucrose. And from the source, always the nutrients are getting transported. So it can mobilize organic compounds, it can capture organic compounds, and where it is source is a place where there is excess photosynthetates are present. So maximum photosynthetates are present here. And again there is, it can always lead to transport out. So sucrose is termed as a translocable sugar. And it is normally, example of this is a mature leaf, a mature photosynthesizing leaf. or even a storage organ. So, place of manufacture is primarily termed as source, whereas when you come to the sink, a sink is always a non-photosynthetic organ. You could also call it immature leaves or developing fruits, which require the food. So, they do not have, there is always a deficit of photosynthetase, they do not have enough of photosynthetates. So, there is deficit of food. And if you take roots or if you take tubers which are growing, so mostly roots act as sinks. So, any relationship between a source and a sink via flow of water through an osmotic gradient establishes a source sink relationship. Now, what really happens at the source? Because this is a very important concept in translocation of organic substances. In the source, there is active loading, that is sucrose molecules get attached or moved to the phloem. So, when you take the phloem, in the phloem we have the sieve cells and also the companion cells. We also have the sieve elements. If you look, these are primarily the sort of a sieve cells and these are the companion cells. So, these are elements of the phloem, and again we have the sieve plate, and we have at the peripheral we have the plasma proteins. Now, all of these are involved in at the source. So, at the source there is build-up of carbohydrates or build-up of energy or build-up of sugars, and sugars are built up because it is an energy driven phloem loading. So, this is termed as phloem loading, and it requires ADP. So, when you take the cells, in these particular cells always the solute potential is low. There is high solute concentration. Solute potential, capability of the solute to move or work is low, but it has got high solute potential. Water potential decreases. The reason why the water potential decreases is primarily water does not have the freedom to move, and water flow is primarily from cell to cell, that is it is based on diffusion pressure deficit. Water movement from one phloem cell to another phloem cell primarily is from cell to cell. So, water enters sieve cells or sieve tubes from the xylem. So, from the xylem we have water entering into the phloem. So, once it enters, then there is high turgor pressure. So, once there is more of turgidity, there is increased hydrostatic pressure. So, an increased hydrostatic pressure leads to loading into the sieve tubes. We have high hydrostatic pressure all round. So, it results in loading up the sieve tubes with relation to sucrose and other sort of solutes. So, how do solutes and sugars get loaded into the sort of a phloem? This is primarily because of high turgor pressure or high hydrostatic pressure. So, that is a very important sort of a concept. Now, we come to the sink. In the sink, which is present at the bottom, the sugar is unloaded. Sugars are unloaded. It means it is also termed as phloem unloading. So, low sugar concentration is present here. And again, because there is low sugar concentration, the solute potential is very high. Parallel to this, water leaves the phloem. There is efflux of water. When water leaves the phloem, it enters the sink. So, once it enters the sink, the phloem turgor in the phloem because water is moved out, the turgor decreases. So, because water turgor decreases, the water potential is lower outside the cell. Is lower on the outside of the cell. So, we have primarily that there is unloading of the sucrose. Again there is osmotic loss of water, minus H2O minus sucrose occurs here, and the hydrostatic pressure is released. Thereby the xylem recycles the water from the source to the sink. There is a sort of an entry or there is a movement of water. So, what do you primarily understand by unloading? Unloading means sucrose and solutes out of the sieve tubes at the sink. So, when we have all the sucrose and the solutes, when they come out, this primary process is termed as sink or phloem unloading. Concept, we are trying to understand absorption of ions, active transport, source and sink relationships. Now, we come to certain important basic concepts. For the first of the concept is termed as bulk flow. What exactly is the meaning of bulk flow? When more water moves in phloem, if you take the phloem tissues, we are talking about water movement in the phloem tissues. And when water moves into the phloem tissues, we understand there is a buildup of pressure. So, because there is a buildup of pressure, it is also termed as pressure flow. So, in pressure flow, there is both the phloem and the xylem are coupled in an osmotic system. We have the phloem, then we have the xylem. It acts like an osmoticum and they are linked, that is, they are coupled. So, there is transport of both sucrose, there is transport of sucrose as well as there is circulation of water. So, whenever there is a circulation of water due to an osmoticum, there is a link between the xylem and the phloem and there is also movement of sucrose. The entire process, which is determined by pressure or pressure flow, is termed as bulk flow. This is very common in green plants. Now, in bulk flow, we understand that flow occurs at the same rate and there is no membrane barrier. So, bulk flow is predominantly driven by pressure gradient. There is non-involvement of membrane in the case of the bulk flow. Hence, now that was primarily about bulk flow. Next, we go into long distance transport. When we take long distance transport, that is, we are talking in terms of translocation of both water and nutrients from the source to the sink. So, from the source to the sink, because of build-up of a pressure, when you take pressure in to pressure out on long distance from a source to a sink. This is termed as long distance transport. So, in long distance transport, there is a translocation. There is a translocation of organic solutes. So, this is very, very important. When we are dealing with relation to long distance, the most important concept is what are we dealing with? We are dealing with relation to translocation of solutes. And what it is based on? It is based on a relationship between the source as well as the sink, which is determined by the pressure. So, whatever we are primarily dealt with, like source sink relationship, long distance, osmoticum, active transport, carrier concept, these constitute the basis for many of the sort of multiple choice questions which occur in the chapters of Ascent of SAP, Transpiration and Translocation of Organic Solutes.